So I'm Anthony Duval. I'm a master's student in Sébastien Rodrigues' lab in Université de Sherbrooke. I'm going to talk about the transcriptomic landscape of Mesoplasma florum in a semi-defined media. So from a systems biology point of view, Mesoplasma florum is just as interesting as the most molecules. It has a tiny genome with a simple metabolism, which makes it easier to model. It clusters in the spiroplasma clade with Mycoplasma mycoides and Mycoplasma capricolum. So they're closely related, and they can they like John said they can be both Mycoplasma and mycoides can be its genome can be transplanted in capricolum. Um, they have the typical fried egg morphology colony we can see on the top right that Mycoplasmas have. Um, so contrary to most molecules, Mesoplasma florum is non-pathogenic, and this is including in its natural host insects. Um, so since its host is insects, it also cannot grow over 34 degree, which means it cannot infect mammals, and in some way it's naturally biocontained that way. It has a very tiny genome, actually, under 800 kb, with only 685 coding genes, which is less than Mycoplasma mycoides. But I think the key point here is its doubling time. It has a very short doubling time of 31 minutes for a molecule. This is pretty short. So it makes it just very convenient to work with. So molecules evolved from gram-positive bacteria, and they experienced massive genome reduction throughout their evolution, which translated into the loss of metabolic capacities. This was enabled by having a parasitic lifestyle with a host. And the host nutrients are varied and plenty, which makes mycoplasmas expert scavengers. They mostly rely on importing macromolecules from the environment. For example, like we saw in the previous talk, they cannot synthesize their lipids, so they'll take what's available. So because of this high nutrition needs, molecules are often grown in rich and undefined media, so complex medium with animal-sourced ingredients. The one that's traditionally used for Mesoplasma is ATCC 1161. It has yeast extract, horse serum, and heart infusion broth. It's very similar to SP4 medium. Horse serum here is quite problematic because it's inconsistent in formulation in different batches. It's also getting more expensive, but all these complex ingredients are mostly inconsistent. So we want to develop a defined media with defined ingredients. So having defined ingredients enables customization of medium composition. In turn, this composition gives us novel growing conditions in which omics data is gathered. In the long term, the integration of these omics data sets, so transcriptomic, proteomic, metabolomic, etc., will improve cell modeling and predictability. So here we develop CMRL-AT, a semi-defined medium for Mesoplasma florum. So we use CMRL 1066, which is mammalian cell culture base. It has vitamins, amino acids, salts, and other micronutrients, and it's chemically defined. It's used in other defined media for molecules, or semi-defined media, I should say. Our lipid source here is Albumax-2, which is purified lipids in bovine albumin mixture. Here, the exact species and quantity of each lipid molecule is approximate, and a document is released per batch, but it's not entirely defined. For the oligopeptide needs of Mesoplasma, we use tryptone, which is a low carbohydrate peptone. It's generally challenging to have a completely defined medium in for mycoplasmas because all mycoplasmas need peptone to grow, and there exists no chemically defined peptone. So I think the hardest part to define is the oligopeptide source. And then we grew Mesoplasma in all metabolizable sugars. So glucose, fructose, sucrose, trehalose, maltose, and mannose separately, and with all the sugars mixed in. So here, essentially, we grew Mesoplasma florum in different CMRL-AT semi-defined conditions with different carbon sources, and we did RNA-seq on them and growth kinetics. Um, I'll just talk about the growth kinetics and the RNA-seq, but we also grew mutants library in these same condition, and my colleague Simon will talk about it in the next talk. So some key points about this new media that performs very well. So it reaches the same density, cell density as ATCC 1161 for glucose. So we got here, it's about 5 times 10 to the power of 9, which is pretty saturated. Other carbon sources yield slightly less cell densities. Also, I think the key point here is that the growth ratio, so the fold change between growth when there's no sugar and when there's the optimal amount of sugar, this fold change reaches 4,000 when we add glucose to the CMRL-AT media. So I think we can effectively say that we are able to change the carbon source. For ATCC 1161 previously, when we actually we don't add the sugar to the media that we normally do, we get a little bit more growth. So that's why the ratio is negative here. It's under 1. So we also did high definition growth curves for every condition. So in blue, we have flow cytometry, so it counts live and dead cells. And in red, we have CFU, so viable cells. And in black, that's the optic density that's of a 560 nanometer wavelength that falls when there's growth due to the phenol red in the media. So here the key points is that the growth saturation again is the same density, and also it grows quite fast in the defined media. It takes 20 hours for to do the full exponential phase, whereas in the complex media it takes 16.5 hours. So here's the conditions in which we did the transcriptome and the visualization of these in the hierarchical clustering. So starting with the group at the bottom. Yeah, so starting with the group at the bottom, we have a CMRL in different sugars. And oops,
maybe I just provide with one here and say that I have to leave.
I've been interrupted here. Yeah. That's fine. All right. So CMRL AT defined media grown with different sugars have mostly the same transcriptomic profile. That's the box we see at the bottom. And except for mannose, which yields a completely different transcriptomic profile, and this remains unexplained yet. Although for ATCC with all sugars, we see a different transcriptomic pattern, and this was to be expected. So here's a volcano plot of CMRL AT with all sugars inside versus ATCC with all sugars as well. So the only difference should be the rest of the media. The genes implicating sugar shouldn't change so much, shouldn't be so differentially expressed. So here we have 109 genes upregulated in ATCC versus 107 in CMRL-AT. So this is a major transcriptomic shift. This represents about 30% of the genome. So I took the top 20 top hits for both upregulated in CMRL and ATCC, and I looked at what the function of these genes were. So in the ATCC and genes upregulated in ATCC, we see a lot of genes that are implicated in purine and pyrimidine metabolism. Surprisingly, I think differential expression here reflects the response to stress in each media. So I think there might be a purine and pyrimidine deficiency essentially in the ATCC media, in the complex media, where the nucleotides come from the yeast extract mostly. And interestingly, in the CMRL-AT, most genes that were upregulated were associated with transport. And if we have a look into these, if we take these top 3 genes here, these are transport genes for putrescin, spermidine, and spermidine as well. So maybe there's a road for medium optimization where adding these components will make a better media, a more performant media. So here I compare the defined media with glucose versus fructose. So as expected, there's way less genes that are differentially expressed. There's 15 upregulated in glucose and 15 upregulated in fructose. So doing the same kind of analysis here, the function analysis, we can see here that in both cases, carbohydrate metabolism genes and PTS genes are the genes that are differentially expressed. And I think this could be useful to refine the annotation of certain genes and identify a certain specificity for some PTS genes, for example. And also, if we take the most upregulated gene in glucose, for example, PEG 622, and we look at the absolute transcript in the bottom left, we can see, for example, that this PTS gene is upregulated, massively upregulated in glucose, maltose, and mannose. So here, gene function annotation could be refined because RefSec and RAST have different prediction, for example. So we have sucrose unknown, then glucose PTS transporter, and hypothetical protein for Patrick. So here, I think probably the substrate is for sure glucose, probably mannose and maltose too. This could be also an interesting way to identify inducible promoters, maybe glucose promoters that can be induced with glucose, for example. Also, this media enabled us to do a qPCR assay to quantify cells. This wasn't possible in ATCC media because animal components in the media interfered with the PCR step. And we can very accurately and rapidly quantify the number of cells, which enables us to do more high throughput growth curve experiments. And it's very accurate for all cell concentrations from 10 to the power of 4 to 10 to the power of 9, which is not shown, but you'll have to take my word for it. The primers I use to amplify are housekeeping genes. So RPOB in this case. So thanks for listening. I was super happy to be part of this experience. Thanks to you guys for listening. Thanks my lab. Thanks to John Glass. And I'm the one in the back left. I missed picture day that day. Shame on me. Do you guys have any questions? I can't hear you, John. You seem to be muted.
Questions?
Yes.
Questions?
I have a small one, actually. I think it's a bit niche, but to show your growth rate. And actually, when I grow Mesoplasma on SP4, with defined media as well, defined lipid diets as well, I see them dying pretty quickly. But here in your semi-defined media, they're staying alive for pretty long. Do you have any clue or just any guess to why that might be?
Okay, so in ATCC 1161 on the left here, that's with 4% sucrose added. If you don't add the sucrose, you just take the bacteria will take the sugar that's present in the horse serum simply. They'll have a stationary phase where cells are viable for more than 12 hours. So the sugar concentration seems to have a major impact on cell viability.
That's very interesting. Thank you.
My pleasure.
So, Anthony, have you, I'm guessing you have, in a paper that we published with the Luthi Schulten group in Cell in 2021, Kim Wise had presented his defined media for the minimal cell and for Mycoplasma mycoides, which I know that's being used by the Sayans group. And he did some experiments by altering the amount of phosphate that he added. And the amount of phosphate, so too much or too little, had a significant effect on growth rate. That might be something that you look at that paper. So this is in addition to CMRL, as I understand.
Yeah, absolutely. I'll look into that. Great.
And then in the real world, does Mesoplasma encounter all of these different sugars?
I think it does because it's theoreticized to be an endosymbiont in the gut lumen of insects. So I think all types of sugars are flowing through the intestine of these critters.
Does Mesoplasma also infect the host plant?
I believe it does not, but the reason I put plant host on my PowerPoint is because it was believed so initially, because that's where it was isolated on the lemon tree, right? Yeah.
Okay. All right. Thank you.
Thanks.